Good morning. Thank you for the opportunity to be here to share with you some of the thoughts about interventions and food safety for beef in the U.S. This is my second trip to Uruguay, and I really appreciate the opportunity to be back this year. I had a wonderful visit a few years ago talking about the same topic and visiting with various people in the country and various aspects of the industry. So what I'd like to do today is to first talk about some of the significant events that have affected safety for beef in the U.S., and then talk a little bit more specifically about post-harvest interventions, and then some pre-harvest interventions, and then a summary of what these might mean for ensuring the safety of beef. So first of all, I'd like to give you a little bit of information about where it all started and how we got to where we are today relative to safety in the U.S. beef. So it all started with an outbreak of E. coli 0157H7 in 1992-1993. And this was really the key event that increased the effort towards food safety in beef in the U.S. And this outbreak occurred in hamburger patties from a fast food restaurant, Jack in the Box, that were not fully cooked and resulted in many illnesses and a few deaths. So this was really the start of really a very intense effort towards safety in beef in the U.S. This is a list of other events that have occurred over the years that have been very important to the safety of beef. After the outbreak in 1992-1993, the regulation of safety in beef became more intense, and there were a number of activities. The first was considering E. coli 0157H7 to be an adulterant. So that means that product that would test positive for this pathogen would not be allowed to go into commerce. I don't have time to go through in detail all of these activities, but some of these have been very important. If we think about some of the things in regulation that occurred, the hazard analysis and critical control point regulation was a key event that established an increase in regulation towards the pathogen with the idea of reducing the risk that it would end up in the final product. The industry began a number of activities that would be related to increasing the safety of beef. They formed some task forces and councils. They started a lot more discussion related to safety. One of the key events was that the presidents of the companies decided that they would consider food safety to be non-competitive. And so they would not keep secrets about discoveries that they made that would increase safety. They would share that information because they realized that if there were outbreaks related to beef, then not only the company involved, but all of the companies would be affected by that. And so they made that decision that I think was very important. There were some other activities. The information that most of the contamination was coming from the hide was a very important development, and the subsequent development of hide wash interventions to clean the hide before the hide was opened is one of the most important events. And then more recently, there have been a number of things that have happened. The discovery that lymph nodes can have salmonella contained in them, and it might be a source of salmonella that ends up in the ground beef. And more recently, the regulation by the government is starting to get more focus away from the E. coli and 0157H7 and becoming more emphasis on salmonella, and the agency is starting to make plans for how that they can make improvement relative to the occurrence of salmonella in meat products. This is a table that just shows the results of the Food Safety Inspection Service testing results for ground beef relative to E. coli 0157H7. And there have been a number of changes that have occurred in methodology and changes that have occurred in the way that the numbers are reported. So it's difficult to make direct comparisons through all of these throughout the years. But I think you'll have to just trust me when I tell you that, that the numbers have been going down as far as positive tests in ground beef for 0157H7, and that the implementation of interventions in the processing plants and a number of other events that I'll discuss a little bit more have led to this decrease in the positives. This table shows the Center for Disease Control and Prevention data on the number of illnesses that would be related either to 0157 or to salmonella. And you can see that the numbers have declined through the years for 0157H7 down to about 1 incidence per 100,000 population. But if you look at the salmonella numbers, you see that as you go through the years, there's been very little change, and that these numbers are considerably higher than for E. coli. And I think this is the primary reason that the regulatory agencies are starting to consider salmonella more seriously now. So there have been a number of things that I think have led to the success in reducing the risk for 0157H7. There have been increased regulatory requirements by the government, the implementation of numerous research results, particularly on interventions and how and where contamination comes from. The industry certainly has placed more focus on key activities and best practices for sanitary dressing, and sharing that information widely across the industry to improve practices for the whole industry. So if we look at more specifically some post-harvest interventions and some of the activities that have improved the safety of beef, this is a list of some of the items that are implemented by companies. I've highlighted ones that I think are particularly important, and I mentioned that the importance of discovery that hide is the primary source of contamination, and that good sanitary practices for removing that hide without transferring any contamination to the carcass is very important. It's easier to avoid contaminating the carcass than it is to clean it afterwards. Steam vacuum is another technology that has been implemented widely. I'll show some examples of that in a moment. As soon as the hide is removed, then to have a carcass wash as soon as possible to remove any contamination is another step that's important. Certainly trimming with a knife any visible contamination that is there on the carcass. And then a final carcass wash that generally includes a thermal intervention. That might be hot water, steam pasteurization cabinet, and in some cases following that with other chemical interventions and, in addition to the thermal intervention. So I mentioned a number of studies have indicated that the hide was the primary source of the pathogens that might get on the carcass. Maybe the definitive study was one showing that dehairing of the hide would result in a dramatic improvement in the cleanliness of the resulting carcass. Just to show you that data, the dehairing was a process that looked very promising. From a food safety perspective, it was very successful. You can see that the hide comes in with a fairly high level of 0157H7 without dehairing the carcasses immediately after, before any interventions have been applied, would have a significant amount of contamination. But the carcasses that had been dehaired before the hide was removed, and so they were much cleaner than very little contamination then resulted in those carcasses. So this was really the data that convinced us that, that a hide wash cabinet was maybe a very important intervention that needed to be implemented. This slide shows some alternative approaches to using a hide wash cabinet for cleaning the hide. The de-hairing process, for reasons that we don't have time to discuss today, was not a long-term feasible process, but the concept of cleaning the hide before opening and starting the skinning process was very important. And these are just a number of compounds that might be used in a hide wash cabinet to clean that hide as much as possible before you start the skinning process. And that might be followed by rinses with either water or chlorine solution, and then a vacuuming step or some other step to remove the excess water so that it didn't run into the freshly opened carcass when the skinning process started. And so if we have carcasses that are fairly heavily contaminated, this was one of the first hide wash cabinets that was implemented. It's a very elaborate, very expensive cabinet that does a very good job of cleaning the hide, so that when they start the skinning process, you would have a significant reduction in the amount of contamination or the risk of transferring contamination to the carcass. This just shows a picture of a carcass after coming through the hide wash cabinet, and then vacuuming the excess water from the pattern lines where the first knife cuts will be made. This is just a little bit of data to show the impact of that. And so we see, if you look at the numbers for 0157 here, if the hides before the cabinet were 44% positive for 0157H7 after the cabinet, then we see that 16% were positive, and the carcasses only 2%, as compared to if there was no hide intervention, we see 17% of the carcasses were contaminated. So a dramatic improvement using this hide wash cabinet. This expensive cabinet was not feasible for all companies. Smaller companies might not be able to afford this technology. We evaluated less sophisticated, less expensive approaches to hide wash system that could be applied at the same time, and was primarily a water wash system, and then followed with a chlorine rinse. And this approach, although not the same effect on prevalence of pathogens, so we only saw a small reduction in the incidence of the pathogen, but what we did see is that there was a large decrease in the level of the pathogens on the hide, so that this is really the data that made it clear that what was most important probably was to get the levels on the hide as low as possible, not necessarily the goal of trying to get zero pathogens on the hide. So during the slaughter process, it was clear that if the hide was a problem, that we needed to keep hide on side and hide off side, or the clean and the dirty side, separate, not only with physical a wall between the two, but paying close attention to where the workers were moving and the airflow between these two sides. Another important aspect was a greater emphasis on the best practices by the employees, to have the employees well trained for sanitary hide removal, and then to, very soon after hide removal, to apply a step, an intervention step, so that if there had been any contamination transferred to the carcass, then you can remove that as soon as possible before pathogens have a chance to attach and become more difficult to remove. So these are just pictures showing the initial steps of removing the hide, and making those pattern line cuts, and why you can clearly see how it would be important to be very careful in this process. This is just some data to emphasize that point and to show that sanitary hide removal is very important to the cleanliness of the carcass. On the left is one plant, on the right is a second plant. If you look at the red line, then that shows the level of these black bars, which is the level of contamination on the hide coming into the plant. And if you look below that to these small dots, these small black dots are the number of carcasses that became contaminated or positive for 0157. And what this shows is that even though the level of contamination coming in on the hide is about the same in the two plants, the carcass contamination is much different. And so one plant was doing a much better job of sanitary hide removal than the other plant. So that can be very important not to get that contamination on the carcass in the first place and have to try and put steps in to remove it. On the hide on side, again, spot cleaning as soon as possible after the hide is removed for any contamination that may have been transferred to the carcass. Could be knife trimming. The steam vacuum has been widely implemented for this purpose. A hind leg steam boot, once the hind leg is skinned, then this goes around the leg and applies hot water and steam to remove any contamination. Another important consideration is cleanliness of the knives and implementing the process of alternating knives so that you make one cut and then you have a clean knife. You put the old one in the sterilizer, you get a new knife out of the sterilizer for every cut, and so that you never transfer contamination from an initial dirty knife. The steam vacuum is used for spot cleaning. It's not a whole carcass treatment. It's for when you can see contamination on the carcass from the hide, and so you can clean that spot, and generally is focused on the pattern lines where the initial cuts in the hide are made. A pre-evisceration wash or a wash that's immediately after removing the hide before any interventions have been applied is also one of the important steps to make sure that if there has been any contamination transferred to the carcass, then that is removed as soon as possible. The most effective intervention for this step is generally hot water, but hot water is expensive, so some companies would use a chemical such as bromine or lactic acid. These are pictures of pre-evisceration cabinet for carcass wash immediately after hide removal to see how that process is conducted. And then on the hide off side, maybe one of the most important steps then is that final wash that is applied to the carcass. Generally a thermal treatment of hot water or steam pasteurization is applied and is the most effective. The concept would be to get the surface temperature of the carcass to greater than 71 for at least 10 seconds to kill any pathogens that might be on the carcass. There are also products that have been removed from the carcass before the final carcass wash. So products like the head, the cheek meat, the wheezan or esophagus that have not been treated by the final carcass wash then might need additional washing or cleaning steps to ensure that they are clean. Other interventions that might be applied after the carcass is chilled and it steps after that. The spray chill that is applied in the chilling cooler to reduce the temperature of the carcass often has a chemical or an antimicrobial of some kind to increase the safety during the chilling process. Various points after that, after the carcass is chilled, you might have an intervention. The cuts before they are packaged might be treated with an antimicrobial, and the trim might be treated with an antimicrobial before it's ground for ground beef. This slide just shows a summary of the relative effectiveness of different interventions that might be applied either to carcass or different cuts or products. And I have E-beam listed over here on the first side, with the highest reduction of the pathogen. But irradiation is perceived to be a problem for most consumers in the U.S., and various petitions to apply this process to whole carcasses without having to label it as irradiated have not been successful. So I don't think that process probably has much hope for use in the U.S. There is a poster that you'll hear about later at this meeting discussing irradiation of trim, and perhaps that it's certainly an effective process, and if it has consumer acceptance, then I think it could be very helpful. Although hot water is a little bit further down the list on this chart, I think it's one of the most effective interventions that you can use on a carcass or on a cut or even heads. So although it's very expensive, and so companies can have selected various combinations of these interventions based on cost and effectiveness and what works best in their food safety system. Another step that I would consider an intervention is this test and hold process that many companies have implemented, where that they take a lot of trim. In this case, it might be 5 combo lots of 2,000 pounds each, or maybe a single combo, and test that for 0157H7 and not release that product into commerce until the test comes back negative. And so this has been very useful to prevent product that's positive for the pathogen going out into commerce. So just to summarize, post-harvest interventions, companies are definitely using a multiple hurdle approach, combining a number of different interventions at different steps in the process, because there's no one intervention that will be 100% effective. A focus on hide intervention to get it as clean as possible, and then the hide removal steps are some of the most important considerations. There are a number of antimicrobial compounds that can be used that have been shown to be effective. There are numerous ones being evaluated up to today. And one of the important considerations is that looking at potential interventions that are non-thermal due to the cost of thermal interventions and that don't require water usage as we try to move towards greater conservation of water. And I talked about the irradiation issue in the U.S. with consumers. So if we turn to pre-harvest interventions, I was advised that I should put pre-harvest interventions first and then talk about post-harvest interventions, but I wanted to talk about post-harvest interventions first because I felt like that they have been much more successful, much more widely implemented. So I'll leave pre-harvest for here at the end. The question that I think you ask is, given the success of post-harvest interventions, why do we need to worry about interventions for the live animal? And I think there are a couple of reasons we should be considering trying to find pre-harvest interventions. So the first one is that we still have some incidents of pathogens getting through the system even with all the post-harvest interventions that are used. So we still need to try to make more improvement. And the other issue that pre-harvest could be very helpful with is if we think that super shedder animals that are shedding a very high level of the pathogen in their feces are contributing to most of the contamination, then it seems logical that a pre-harvest intervention that was effective might be the best approach to to eliminate or reduce the level of animals that are super shedders. And certainly we should consider that there's environmental contamination, both from the dust and from runoff that might come from feedlots and might be contaminating the environment, might be leading to contamination of produce that is grown in close proximity to feedlots. So if we can reduce the levels in the live animal, then we can help that risk of that happening. And also there's the idea that the live animal production side also should share in the cost of controlling the pathogen and not have all of that burden land onto the processing plants. So this is a cartoon to show that if we don't have pre-harvest interventions and we're relying solely on the post-harvest, and we have a multi-hurdle approach, we have most of these post-harvest interventions in place, as long as the incoming level on the hide is not too great, then this will be successful, and we can control the pathogen and not have the pathogens escaping into the final product and reaching the consumer. But if that level coming in is higher and maybe too high and overcomes the ability of the post-harvest interventions to eliminate all of the contamination, then this is where we might have a problem. And so I mentioned super shedders, and so I think that one of the ways that we might have this situation where the incoming level is too high for the post-harvest interventions to control is when we have a super shedder animal in a pen. And this is a figure that shows, if you look at these black circles here, these are pens that have at least one super shedder in them. And so we have both a high 100% prevalence on the hides of the animals in these pens, but more importantly, we also see that these are the pens that have the high levels. Not only the pathogen is present on the hide, but it's present at a very high level, and so we have a much higher risk of transferring that contamination to the carcass at harvest. So if we did have an effective pre-harvest intervention, then we can reduce those levels on the hide coming in, and perhaps we can reduce them to even a very low level, and then the likelihood that the post-harvest interventions will all be successful and that we will not have any contamination on any final products will reduce the risk then to the consumers. One of the problems with a pre-harvest intervention is the concept that a lot of different animals are coming to the processing plant, and they come to the holding pens and all the facilities at the processing plant. And if you do have contamination coming in, then all the other animals that come after that then are very likely to become contaminated. And so if we do have a pre-harvest intervention, it will be most successful if it's widely adopted so that the animals are cleaner coming in and we don't have this buildup of contamination at the lairage facility at the processing plant that can contaminate the hides of all of the animals, even the ones that have been through a successful pre-harvest intervention. And this is just to illustrate the impact of the lairage environment at the processing plant. And so this table shows for both E. coli 157H7 and for Salmonella on the carcass immediately after removal of the hide. And we see that we were tracking the genetic types of 0157 and showed that the ones on the carcass, 75% of those came from the lairage environment, and 84% of the Salmonella positive carcasses, those Salmonella came from the lairage environment. So this is an important source of contamination of the hide that we have to consider when we start thinking about a pre-harvest intervention. These are some of the options for pre-harvest interventions that have been studied. And so direct-fed microbials or probiotics, Bovamine is the one that is most widely used. It's fairly widely used in the U.S. It doesn't actually have a claim against pathogens, but a number of studies have shown that it's effective against O157H7, and so it has been adopted by the industry. Sodium chlorate is another compound that has been studied. The initial experiments show that it appears to be very effective, but it has not obtained FDA approval for use in animals that would be used for food. So, and it's actually been waiting for about 10 years at FDA to obtain this approval. So, unless that approval becomes effective and so that additional studies can be done, then we won't know for sure whether in commercial practice that sodium chlorate would work. Vaccines are another technology that have been studied fairly widely. This first one from Epitopics has a conditional license in the U.S. The initial smaller trials showed it to be fairly effective, but it had less success when they did large commercial trials. And I think largely because of the cost-benefit trade-off, it hasn't been adopted widely. Icomst is another one from Canada. It's fully licensed in Canada, but not in the U.S. And has had similar results in trials. The Salmonella Newport vaccine also has conditional license in the U.S., but it's mostly used by the dairy industry. So it might be useful for those harvesting cull dairy cows to reduce the incidence of salmonella. Bacteriophage is another potential control for pre-harvest. It has been adopted in the U.S. by some companies. They spray the phage on the hides at the processing plant after they're unloaded before they go to the lairage in the holding pens. So generally during the warm months of the year when they have the most issues with O157, then they would be spraying the hides with this product. They're also working on expanding the product so that it would have effectiveness against the other 6 E. coli serotypes that have been adulterants in the U.S. There's also these best practices that seem like a really good idea. Clean feed, clean water, maintain the pens well, reduce insects as much as possible. But there's really no data that shows that these would have much of an impact on the pathogen. So how effective would a pre-harvest pathogen need to be useful? First of all, very few of them have been shown to have strong efficacy. The most widely adopted is the Bovamine probiotic. The bacteriophage for O157 has been implemented to a lesser extent. Most of the reason for the Bovamine probiotic implementation, I think, is because there's some growth efficiency benefit that offsets the cost, and so the food safety benefit is kind of a bonus. But for most of the others, the cost benefit I don't think is perceived to be favorable yet because the cost is just too high for the effectiveness of the intervention. And I think the real answer to how effective do they have to be is that if they reduce the super shedder animals to a low level and not necessarily prevent create 0% pathogen in the cattle, but at least eliminate the ones that are shedding at very high levels, then I think that would be useful, very useful to the industry. And then the issue becomes the cost. So in conclusion, there's no silver bullet. There's no one intervention that's going to solve the problem. So The approach has been to use multiple interventions and integrate that scheme throughout the process to obtain the maximum reduction in the risk of pathogen ending up in the final product. So there's sequential interventions that complement one another. The focus is on the hide, not only cleaning the hide, but the process of removing the hide so that you minimize contamination on the carcass. Post-harvest interventions can and have been very successful, but I think clearly to make additional progress, effective pre-harvest interventions are needed. And I think this will require that we improve our current pre-harvest technologies, or maybe obtain new knowledge, such as a better understanding of why some animals are super shedders, so that we can devise a strategy to eliminate those animals shedding very high levels. And I think the bottom line is that to produce safe beef, companies have to have the right food safety culture. It's developed from the top-down commitment that includes the right attitude, the right tools and training, so that the employees have the ability and the desire to do the right thing all the time, every time, day in, day out, to make sure safe food goes out to consumers. And hopefully we'll make more progress on pre-harvest and post-harvest interventions that will allow companies to do this. I would like to acknowledge my colleagues at the Meat Animal Research Center. A lot of them have contributed to our efforts, and also to these 4 individuals that contributed data or slides to this presentation: Mindy Brashears and Guy Lonergan at Texas Tech, Todd Calloway with USDA, and Mohammed Kamari with IEH. Thank you very much. It's been my pleasure to be here. If you have any questions.